possible. We assume matings are random, and that may or may not be true, but we're going to make that assumption. We assume mutations do not alter the frequencies of the alleles, and we'll talk about mutants and mutagenesis a little bit in this lecture, but that's a pretty safe assumption. And we're going to assume there's no migration of populations that have different allele frequencies. We're not mixing and matching populations, and we'll treat that separately. And we're going to assume there's no selection for one allele or another allele. And if all those things are basically true, or we can regard them as true, then we can apply Hardy-Weinberg. Okay, so good. That just summarizes kind of what we had talked about before, and then we looked at this graph and so forth. So what we're going to do is look at a few really straightforward problems where we can apply the Hardy-Weinberg equation to start to answer interesting questions in population genetics and pedigree analysis and so forth. So the first problem, and these are from the book, so if you want to review them, they're in the chapter. But the first problem is concerning a recessive genetic disorder. In this case, it's cystic fibrosis. It's an autosomal recessive disorder, and we're going to make or we're given this information that 1 out of 2,500 people, at least of European descent, have cystic fibrosis. So this isn't something we dream up. This is a number or a fact that we suppose is accurate. So we're going to use this 1 out of 2,500 number and try to make some conclusions about the population that gives rise to 1 out of 2,500. So 1 out of 2,500 is 4 out of 10,000, so this is a fraction. 0.0004 of this population are of the genotype recessive allele to cause this particular trait. Okay? And we know in that Hardy-Weinberg equation, if P is the frequency of the dominant or wild-type allele and Q is the frequency of the recessive allele, we know from Hardy-Weinberg that Q squared is the term that equals the genotype frequency of people that have 2 copies of the recessive allele. So we can say that Q squared equals this value that we've been given in this problem, right? And then it's obviously pretty straightforward to determine that Q, the allele frequency of the cystic fibrosis causing allele, is 0.02 in this population. And that's an interesting number. I mean, that's 2%, right? I mean, it looks like something minuscule, but there's a value to it. And it's kind of hard to get your mind around this notion, but nonetheless, that's the value that we observe or we calculate using Hardy-Weinberg with this number that we start with. And we know that if there's only 2 alleles, they both have to add up to a total of 1 or 100%. And so we can easily calculate P if we know the other one. And so P is 0.98. So those are pretty straightforward things to calculate. And then we know that that middle term in Hardy-Weinberg, 2PQ, is the genotype frequency of the heterozygotes, which would be carriers of this cystic fibrosis allele. So 2PQ, we just plug in what we've just calculated, and we get a frequency of 4% for carriers of the cystic fibrosis allele, assuming that this 1 out of 2,500 number is accurate. Okay, so again, 4% is a number that's a little bit hard to kind of imagine or envision, but it seems like a lot, right? 4% is seemingly a pretty large proportion of the population of European descent, which are carriers of this particular disorder. And later we'll kind of briefly investigate this notion of well, why in the world can something that is bad like this have a carrier frequency or an allele frequency that's so high? How can we imagine something like that emerging in a population? Okay, so simple problem. We kind of plug and chug using our P and Q is 1, and P squared, 2PQ, Q squared is 1. Here's another one, and it involves X-linked trait. And it's really about the same, but we have to keep something important in mind, and that is for females, X-linked traits will follow pretty much that Hardy-Weinberg set of terms. For a female to have let's say 2 copies of big A allele, it will be P, the frequency or the chance that you would pull out big A on the X chromosome, and you would do it again, right? That would give a female with 2 X chromosomes with that A allele, so that's P squared. Q squared is the same way, the chance of the big B allele in the population, and the chance of getting it twice, which is Q squared, and then the heterozygotes will be 2pq, just as before, because there's 2 chromosomes, we have to pick twice. For males, you don't have to pick twice. You imagine, or you know what the allele frequency in the population is, and you just have to pick once. Each male has 1 X chromosome. So the frequency of getting XA for the male is just putting your hand into the vat one time and pulling out with some frequency P that XA chromosome. And for the XB allele for the male, again, the idea is you just have to pick once. Okay, so that can cause a little bit difference in how you think about these problems when they're X-linked for males and females compared to autosomal disorders. So a really simple example. Yeah, question?
So for males, the equation would just be P plus Q.
Right, pretty much like that, yeah. So the example is pretty simple here. That is X-linked color blindness. If it has an allele frequency of 0.039, this is something we're just given in the problem. We can determine the frequency of affected females, and we can determine the frequency of affected males. So we're given kind of the, this Q value. Females, we have to use this term Q squared. That is 0.039 squared, which turns out to be this fraction of the population of females. We'll have this. For males, it's just Q itself. Okay? And the note I have at the bottom here is just to remind you that it's not double or half. It's not 2Q or half a Q or something like that. And sometimes this can be tricky. What we're doing here is it's squared or not squared. So this is not half of this. When you think about pedigrees, you often think in terms of, well, half the males or some fraction of the males compared to the females and so forth. And in pedigrees, you already know what people are because you see that they're affected somewhere in the family, right? And you can trace down the frequency of an individual, what's the chance they have some allele and so forth. And sometimes in those cases, we do kind of deal with this half males affected, half males not, and so forth. So don't get tricked by the halfness. There's no halfness in this particular style of problem. It's the frequency Q or the frequency of Q squared. Okay. What if there's more than 2 alleles? Well, we can run. That's one possibility. Okay. But another possibility is that we have to try to deal with situations where there's more than 2 alleles, because this is obviously a common situation. And there can be obviously lots of alleles, and it is very complicated. So let's think about a situation where maybe you have A1 allele, A2 allele, A3 allele. You start to have a lot of possibilities for genotype frequencies, and then you'll have at least 3 P, Q, and R values than for the allele frequencies. So in this case, the total sum of all the alleles will be P plus Q plus R. We've just added in another one, and that has to sum up to 100% or a value of 1. So that's not so different. Yes, question?
Let's go over again what R means. I'm sorry.
So P, okay. P is the allele frequency of, let's say, A1 in this example, okay? Q is the allele frequency of another allele, let's say A2. We're just adding in another term, R, to reflect the allele frequency of yet another allele, let's say A3. If you wanted to add more here, you could. That would just mean there were 4 alleles or 5 alleles. So we need three variables here to designate the allele frequency for each allele we're talking about. So in this case, it's P plus Q plus R equals 1, so it's a 3 allele system. And then the question is, well, what does the Hardy-Weinberg equation look like if you have 3 alleles? And then here it is. It's kind of a disaster, but it's actually something that you can understand. It's not too bad. We have a P squared term. That's going to be your chance of getting, let's say, A1, and then your chance of getting A1 again by putting your hand into the vat and picking out A1 allele. So that's P squared. Those individuals would be the genotype frequency of A1, A1. Q squared would be the genotype frequency of A2, A2. R squared, you get the idea. And we've got some other ones over here. So 2PQ would be the genotype frequency of individuals where you picked P, which is A1, and you picked A2 frequency Q, but you can do it two ways, P first then Q, or Q first then P. So there's 2 times that, right? And so you've got some other terms as well that reflect the frequency of the heterozygotes in this 3 allele system. So this is the Hardy-Weinberg equation for 3 alleles. Each of the terms is going to give rise to a genotype frequency of individuals of the various types, for instance, up here, and we set it equal to 1 because everybody's got to add up to 100% of the population. Okay, so how do you get this? If you think about putting your hand into the vat and picking anything, your chance of picking anything is 1, right? And your chance of putting your hand in the vat again and picking just anything is also 1. So 1 times 1 is, 1, right? Well, what does the 1 equal? The 1 equals P plus Q plus R. You put your hand into the vat and you either pick P or you pick Q or you pick R, but you're gonna pick something. And the frequency with which you pick them is given by either P or Q or R, but you pick something. And if you do it again, you're gonna pick something, and that's gonna be either P or Q or R. So your chance of picking something times your chance of picking something is basically PQR times PQR, right? And if you expand that out, you essentially get this equation here. So it kind of makes sense. And the other thing, the last thing I point out is thankfully we're not triploid. So you might ask yourself, what if we were triploid? How complicated would it get in that case if you think about this kind of picking once, picking twice, and now picking a third time, okay? So it would be even worse than this. Okay, so let's look at an example 3 allele problem. And again, this one is straight out of the textbook, so you can refer to that if what I say is confusing. And this is a blood group problem. We already know kind of how the system works. We have 3 alleles, IA, IB, and small I. And we're gonna try to figure out the frequency of these alleles in some population. So at the start of the problem, we're given some information about the frequency of blood types in this population, right? So this is kind of a phenotype that we're given. And we're told type A individuals are 53% of the population, type B individuals are 13%, type O individuals are 26% of the population. Okay, so you know how these genetics problems are. They start you out in some irritating way by omitting something and making you do something right away to fill in what's not given. So the first obvious question is kind of, well, wait a minute, we don't have all of the blood types present in our list here. We have something missing, which is AB, right? And we can easily calculate AB just by taking 1 minus all of this. So whatever's left over with 1 minus all of this is going to be the AB group, right? So we can say AB frequency in this problem is 1 minus the sum of everything else here. So this obviously means that if you look at the frequency of all the possible blood types, A, O, B, AB, they have to add up to a level of 100%. You can't have something missing and you can't have too much or too little. That's why sometimes when you're given some information like this, one thing that you can do is check right away, is it adding up to 1, or is there something missing, or does it add up too much and it doesn't make any sense? So those are things to think about. So we've already answered the first part of this question. What's the frequency of the AB blood group? Okay. So now we want to ask the question, well, based on these phenotype frequencies, what are the allele frequencies of IA, IB, and small I in this particular population, right? So we want to find the frequencies of each of the 3 alleles, and we have this equation that we just looked at where we have these P and Q in our variables, and we're going to assign P as the frequency of the IA allele, Q is the frequency of IB allele, and R is the frequency of the small I allele, right? So fine, that's pretty clear. And we know that we have the Hardy-Weinberg equation for 3 alleles, which is this big mess all written out here. And so we know that P squared is going to be A-type, and it will be A-type with the genotype of IA, right? If P reflects the frequency of IA allele in your P squared, it means you have IA and IA. 2PR will reflect the genotype frequency of individuals that are heterozygous and of type A. So they have P, the frequency of IA, they have R, the frequency of small I, which is kind of inert in this system, and you can do this 2 ways. P then R, or R then P, so it's times 2. So these 2 terms, this one and this one, added up, will give us the frequency of A blood type individuals in the population. So that's pretty straightforward, that makes sense. The problem is we want to now try to determine what the actual P, Q, and R values are in this problem. And so first of all, we're going to look at individuals that are type O. And the reason is obviously there's only one term in the equation that reflects individuals that are type O. If you're type O, you've got to be small I. And that will be given by the chance of picking small I, which is R, times the chance of picking small I again, which is another R. So R squared are these O individuals. So here we go. People of type O, blood type, are genotype small I. In this problem, the frequency of O individuals is 0.26. So 0.26 is the frequency of O individuals. We know that's reflected in this R squared term here. That's how you get 2 copies of small I. And so we can just determine then that allele frequency for small I given by this R term is 0.51. So that's pretty easy. And we've got 1 of 3 of them done now at this point. Okay, but we've got to do IA and IB, and so we'll do IA. The problem with IA is that there's 2 possible genotypes, both of which give rise to a blood type, okay? And so there's 2 terms in that equation that cause the A blood type. The terms in the equation are P squared and 2PR. So we know that the sum of these is the frequency of A blood type individuals, which in this problem was given to us as 0.53. So P squared plus 2PR equals 0.53. That's pretty straightforward, right? We've got this term and this term that we have to add together to get the frequency of these individuals. The book solves this in a cute way. The cute way is to make this simple, they add an R squared to this side, and we already know what the R squared is. We're kind of given that in part of the problem. And so when they add R squared to this side, they add 0.26 to this side. And it's just a little trick that enables them to simplify this side of the equation to be P plus R squared equals some value here. And then we take the square root, P plus R is 0.89, and we already know what R is it R, right? 0.89 minus R equals 0.38, and so we get the value for the frequency of the IA allele, which is given by P, as 0.38, okay? So we get another one, and I should say, or just reiterate what I've said in red here. This is a cute way to solve this, but this isn't an algebra class. So if I try to design problems of this type, I will try to design them so that we're thinking about the genetics and so forth. We're not trying to go back to middle school and figure out these types of things. And you don't have to do quadratic equations and stuff like that. I will design problems so that we don't have to mess with this. This is nice enough, right? But this is just a way to get to the answer that we're interested in. Okay. So we get now allele frequency R, allele frequency P, and we have 1 left, which is Q, which reflects the allele frequency of the IB allele. And we know that the sum of all of these alleles has to equal 1. So P plus Q plus R equals 1. That's the total, allele, set of alleles in the population. And we've already determined P, we've already determined R, so we can just plug them in, and so we get 1 minus some value, and it turns out then that Q is 0.11. Okay, so neat. Blood group problem using Hardy-Weinberg equilibrium to go from, in this case, phenotype frequencies, understanding that phenotypes were generated by multiple genotype possibilities like IA, and IA small I, and then ultimately using Hardy-Weinberg and getting the allele frequencies. So I should also say that for this to work and for the answers to be in any way right, we have to assume the population is in Hardy-Weinberg equilibrium. Okay, so that's an assumption that we have to make here. So conclusion of the problem, population of 53% A, 13% B, 26% O, and 8% AB, which they asked us to calculate, that is in Hardy-Weinberg equilibrium, this is an assumption, will have allele frequencies given by these values, and we've just determined that. It's not so bad, you just kind of see what the point of the problem is, you understand how to use the equation, what the terms reflect, and so forth, and then you just kind of plug and chug it out. All right. Good. So here's an interesting problem. This problem asks the question, is the population in Hardy-Weinberg equilibrium or not? Okay, so our problem here, again from the book, involves these two words, okay, which is a vole. So here's a vole. Isn't it the cutest vole you've ever seen? I think it is. And so we're going to collect voles, and we're going to measure alleles of the transferrin gene. And we have two alleles. They're called M and J. Okay, so this is just stuff we're given, right? This is like the little part of a multiple choice problem where you're given some information and you just have to absorb that. And you say, okay, there's a gene, there's two alleles, and we collect some organisms, right? So what do we collect? We collect a total of 77 voles here, and then we divide them into bins as being genotype MM, MJ, or JJ. So we have to have some way to do this, right? Maybe it doesn't involve just looking at them, maybe there's some molecular approach, but whatever it is, we have some way of saying, of our 77 animals, we have 12 MM, 53 MJ, and 12 JJ. So that's a measured thing, that's a fact, okay? And then we can use one of our allele frequency determination formulas, a clunky one or that kind of one based on genotype frequencies to determine allele frequencies in this population. Okay? And again, this is going to be a fact. The allele frequencies are derived from the things that you've actually measured. So allele frequency P of the big M allele will equal 2 times the number of these individuals plus 1 time the number of the heterozygote individuals divided by the total number of kind of transmissible alleles in this system, which is 77 animals times 2 alleles each. And when we do that, we get allele frequency P for the M allele of 0.5, and then 1 minus that will give us the other allele since it's only 2 alleles. So Q, which is the allele frequency of J, is 1 minus P is also 0.5. And the fact it's 0.5 is, it's kind of one of these made up problems, right? Like there's 1,000 progeny exactly kind of problem, which I don't care for so much. But nonetheless, we get these kind of perfect half and half values for allele frequencies P and Q. All this is measured, it's factual. There's nothing you've done here to interpret anything or guess about anything. Now what we're going to do is take the allele frequencies that we know are correct based on the number of organisms of certain genotypes that we've collected here. We're going to take those allele frequencies, we're going to plug them into the Hardy-Weinberg equation, P squared, 2PQ, and Q squared. So one of the terms is P squared, that's allele frequency P times allele frequency P. That should equal, we suppose, the genotype frequency of MM individuals, and we get P squared is 0.5 to 0.5 is 0.25, right? And then we can say, well, how many animals out of 77 are, 1/4 of them? So 0.25 times 77 gives us about 19 animals predicted to be of this MM genotype. 2PQ is 0.5, we just plug it in. So that means half of the 77 animals are predicted by Hardy-Weinberg to be heterozygotes, and that's about 38 or so. And Q squared is 0.25, so that means 1/4 of our 77 are predicted by Hardy-Weinberg to be homozygous JJ, and that's another 19 here. Okay? And then the book analyzes this through that chi-square analysis, and I have a note here. Not shown, and which you will not have to do again. I will not ask you another chi-square problem ever in this class. So, we're just gonna say, okay, we do it and wave our hands, and then just informally say that, hey, wait a minute, the numbers of organisms that we predict when we plug the real allele frequencies into Hardy-Weinberg here are not matching very well with what we actually observed when we collected these animals. Okay, so that's weird. The chi-square analysis says, hey, this isn't fitting, and we're going to conclude then that it seems like this population is not in Hardy-Weinberg equilibrium. And I have a few comments about how we should think about that. So the results say, when you measure, and this is a fact, when you measure allele frequency P of M allele and allele frequency Q of the J allele in this population, these are facts, right? And you try to plug them into the Hardy-Weinberg equation, you don't get the correct frequencies of individuals of each genotype using that approach. And so now we get this kind of but wait moment. But wait, that seems weird. How can you determine allele frequencies and then get the wrong number of organisms when you plug them into this Hardy-Weinberg equation? Isn't it the case that allele frequencies have to relate in some way to genotype frequencies? And the answer to that question is yes. Allele frequencies do have to determine genotype frequencies, but they don't always have to using that Hardy-Weinberg equation. That equation makes all those assumptions. So in this particular case, we might suppose that this population for which we've measured accurate genotype and allele frequencies, maybe this population is not in Hardy-Weinberg equilibrium for some reason, like migration or selection or something like that. So just keep in mind that you can't automatically take allele frequencies and just plug them into Hardy-Weinberg and always expect to get exactly the right answer. You have to make a decision about whether the conditions apply or don't apply. So here's a population where we try to do that and we're getting the wrong numbers of wrong genotype frequencies and numbers of organisms. Okay, I made another note. An extreme example, you might imagine a population that consists of only 2 genotypes, right? You collect animals and you collect 50 big A, and you collect 50 small a, right? So you know that your allele frequency is 0.5 and 0.5. You plug that into Hardy-Weinberg and you certainly are going to get a big heterozygote category as predicted, but that's not true at all, right? You could imagine a population like this, at least theoretically. So this maybe is an extreme example, but this kind of highlights the notion that just because you calculate these things doesn't mean that you can always plug it back into Hardy-Weinberg. Okay. So how can they not be in Hardy-Weinberg? And I think I've already said this. Population not infinitely large, forget about it. Matings might not be random. There might be mutation, although we're going to talk about that as not being a major factor in deviations. No migration, no selection, those are things that are important that we should think about. So what we're going to do is kind of examine some of these types of topics, mutation, migration, and so forth in the next few sections. So first one is alleles come from mutations, right? This is the source by which nucleotide sequences change from one person to another person or from one generation to another generation and so forth. So how does mutation happen, or what is mutation? Typically you think of a mutation, first of all you think of a mutation as being bad, right? And you also think a mutation is you have a sequence of DNA with a specific nucleotide sequence from one end to another, and you have a change at a given spot, right? So that's a mutation, a single nucleotide sequence change that changes the sequence from one thing to another thing. Maybe it prevents production of that particular protein, for instance. So that's pretty clear as what a mutation is. So when we think about individual sequence changes, how do they arise as a result of mutation? Well, DNA is a stuff, right? It's a material, and the material gets old or has a problem, so chemicals have a certain lifetime. And so there's going to be, intrinsically to DNA, a certain possibility that the chemical falls apart or does something wrong or whatever, right? That's pretty clear. So just due to the fact that it's a substance, substances break. And so the intrinsic chemical properties of DNA are going to, in part, determine how fast sequences mutate. Things like demethylation, deamination, stuff like this. Exposure to damaging agents, mutagens, right? All these labels that say such and such is a carcinogen, and so forth. Those are chemicals typically that are genotoxic. They enter the cell. Many of them go to the nucleus. Many of them like the basis of DNA, and they will add on or link or covalently bond or something like that, and that causes sequence changes. Efficiency of repair mechanisms. The intent of repair mechanisms is usually to make sure the sequence, when it's copied or if it's damaged, gets returned to its original form. But repair mechanisms have a certain accuracy. They're not 100%. Maybe they're 99.9999 whatever percent. They're really good, but they can't catch everything. And it turns out that one explanation for carcinogenesis in some individuals is they have defective DNA repair enzymes, so they tend to accumulate mutations far more rapidly than they ought to, and that results in oncogenic transformation. So this is one way, repair mechanism efficiency, which we can generate more mutations or less mutations. The accuracy or fidelity of DNA replication. DNA replication is accomplished by these DNA polymerases. We'll talk a lot about that later in the course. But these DNA polymerases, they try to be accurate, but they make mistakes. Okay, so that's another source of mutations. These are all typically things that will cause one nucleotide to change to something else, the kind of single point mutation idea. The other things that we've talked about for mutation maybe include recombination errors. That would be enormous mutations where you invert regions of chromosomes or transpose regions, things we saw in those chromosomal abnormality chapter, right? Typically those types of things aren't due to these four things, but they're due to big recombination mistakes. Enzymes find two pieces of DNA, swap information, and you get these huge sequence rearrangements. So the word mutation, typically everybody always thinks it's bad. And in a sense, that's true. If the mutation rate in an organism is too high, the effects are very serious, right? Like carcinogenesis and so forth. And if that's the case, then the species, whatever it is, accumulates mutations far too quickly. The offspring are damaged, or they're carrying this load of mutants that causes proteins to be broken and so forth. So that's not good. You don't want mutation rate to be too high, but you don't want it to be too low. Right? If the mutation rate would be 0, then the organisms would never change, would never adapt, and so forth. So it's, in that sense, mutations are good because they're the way that you generate new sequence variants, new alleles that might have some beneficial effect. So you've got this kind of two-part idea here with mutations. The book goes into a lot of kind of pathway detail or math detail about forward mutation rates and reverse mutation rates and all that stuff. And I don't want to do that. I just want to show a picture of the idea of what they're talking about. What they're saying is that a particular allele, call it big A, has a chance to undergo, let's say, a point mutant to create a new allele called, in this case, small a, right? Typically that would be defective or recessive, perhaps, but sometimes it might provide some beneficial effect. So there's some forward rate, in a sense, where you go from big A allele to a new allele, small a, and there's also a chance that you could go in reverse and revert whatever change you made going in this direction and turn it back into this direction. We're not going to worry so terribly much about this and all the kind of stuff the book describes, but just understand that we can codify this as going this direction and that direction. And that would be true for every nucleotide along the length of the sequence of a gene. A couple points here. To be directly important for heredity, mutations you would think would have to happen in germline cells, right? If you have a mutation in a skin cell, the effects could be bad, but they don't directly affect transmission of alleles during matings and so forth. That would only be affected by having a mutation in the germline. So when you think about mutations, you really want to separate the effects in the germline, which can be transmitted to the next generation, so a new allele is now there in the population, versus a mutation that affects a somatic cell where you could have carcinogenesis or something else bad, but it's not directly being transmitted through these Mendelian types of processes. Okay, so mutations in somatic cells are not inherited, although you could imagine that they would be have an effect on germline transmission. When people look at differences between human beings, they see about 1 in 1,000 nucleotides being different across the whole genome. And that sounds like not very much, but if the genome is 3 billion, then 1,000, 1 out of 1,000 is quite a few changes. The thing about this comparison is that it's not the same position. So if you compare one genome to another genome, and you say, here's the reference genome and here's the second genome, you'll have lots of changes between the two, right? But if you take that reference genome and compare it to another genome, you will also have a similar level of changes, but they will be often at different spots. Okay, so it's not the same 1 out of 1,000, it's different from genome to genome. Okay. Most of these differences, we have no idea what they do. A subset of these nucleotide sequence differences from one person to another result in these, let's say, recessive alleles, defective protein production, and so forth. Okay, so here's a question that you should probably have some idea about, because if you took a genetics class and your parents ask you, something like, why are there bad genes? It's a basic question. So let's kind of think about this: mutations, bad genes, and so forth. Why are there bad genes? This is one possibility, but that's not the case, obviously, right? So what are bad genes? Bad genes are defective alleles, right? And we've looked at lots of examples of these types of things. And the question is, why are they there? Why in the world are they there in the first place? Okay? And there's some basic ideas to explain why they're there. First of all, bad alleles are mutations that cause something to be defective, but we don't see them, right? You don't see them because organisms are diploid, so they're masked. So if a mutation occurs, let's say in the germline, causing a gene to be defective and it's transmitted, you won't see that at all, maybe until the frequency of that bad recessive allele rises in the population to some appreciable extent, where you now start to have individuals that are homozygous recessive. So diploidy hides them. That's a good thing, right? But it's also kind of a bad thing at the same time. Okay, so you don't realize it until it's too late. Yeah, question? Do people with genetic have common ancestors? I think to some point, right, you see it running in pedigrees, so I would say yes, but then you also have to allow for some spontaneity of mutagenesis. So it's a balance between these things. Once it's there, there's a possibility of transmission. So that's kind of, it's in the ancestor and then it's in the offspring kind of thing, right? On the other hand, there can be unexpected things happen mutagenesis-wise where things just happen. So I think it's a combination. You can have mutations happen more than once. You can have them happen twice in multiple families, right? That's possible as well. Okay, another reason why do bad alleles rise in concentration in a population is that they might be helpful to an individual when they're present in a heterozygous state. And one example we looked at before was that sickle cell trait. The idea is some of those hemoglobin variants are thought to potentially confer some resistance maybe to malaria for individuals that are heterozygous, right? So that's a kind of selective advantage for heterozygotes, which will increase the concentration of the recessive allele. But the downside of it is when you increase the recessive allele concentration, you get more people that are homozygous recessive and are affected seriously. Another example is the cystic fibrosis recessive allele. There's a lot of speculation that the reason that it's so high, right, we calculated in another problem, it was like 2%, if those numbers are right, with 4% carriers. Why so high for something that's so serious? And people have speculated that being heterozygous for the sickle cell allele might confer resistance, and I've seen confer resistance to tuberculosis, typhoid, plague. I don't know whether those are right or wrong, but people speculate as to what good these recessive alleles might have. And if there's some good in the case of heterozygotes, you'll see an increase even though they're otherwise bad for homozygous recessive. And the last one, mutations that are bad might be linked to genes that are good. So linkage is important. So here we have a piece of DNA. You've got an allele here that is beneficial to the organism, right? And close by, you have, let's say, a recessive allele. A mutation happened, and this gene is now defective or broken or not as good as it ought to be. Why would this thing increase? It could increase because it's nearby something that has some beneficial effect. So as you kind of increase the allele concentration of the good one, the bad one gets carried along with it because it's so closely linked along the DNA. So linkage could be another potential explanation why you would increase allele frequencies for things that otherwise would just seem to be bad.
So that's maybe if, the mutation, for whatever reason, it's acting on the wrong DNA and one of the vaccines and so forth, that
Uh,
well, what, I mean, it sounds to me like we're talking about really those genes, that are right next to each other are probably making sense for the same mutation, but.
No. I'm thinking about two separate genes altogether, right? One of them is an allelic form that is increasing in concentration through time, generation after generation, because it confers some advantageous effect on the organism and allele transmission. But you could have something that's linked to it that is disadvantageous, but not terribly so. But they're going to go together and be increased together because one is conferring this advantageous effect on the other due to the fact that they're linked together. So it's basically selecting for one thing and something else is kind of piggybacking along, but it's hidden in a sense, but is maybe defective or recessive. Okay, second question that you ought to kind of be able to answer to some extent. Why don't bad genes go away? And this is typically having to do with the fact that we're diploid, right? Usually for these recessive alleles, there's usually no effect on the heterozygote. The heterozygote is a completely normal carrier. So the only way that you might be selected against homozygous recessive alleles is in the form of organisms that are homozygous recessive. Okay, so that makes it very difficult to get rid of these things. And actually in the book, there's some graphs towards the end of this chapter where they show the decline in some recessive allele based on certain hypothetical conditions, the degree of selection against certain genotypes. And you can see that it declines, but it levels out and it becomes very, very difficult to completely ever get rid of these things. Okay, topic 4, some other things that are going to affect allele frequencies. Mutation will affect allele frequencies, but probably not significantly. The idea of mutations is that they're rare chance events. They occur from time to time, but we don't really think of mutations as being a source of kind of wholesale allele frequency changes in a given generation or something like that. Okay, so they're super important because they're the source of new genes or new variants, varieties, and so forth. But they're probably not going to change allele frequencies appreciably in a real fast way. Genetic drift. This is the idea that in relatively small populations, you can have big variations in allele frequencies just through statistical accidents. of who happened to mate with who in a small population and what alleles they carried. And we'll see an example of this. The idea here is if you flip a coin 10 times, you're not quite sure what you're going to expect, but probably not 50-50, and it might be, might not be. Could be 7-3, 6-4, even 8-2, so forth. As you increase this more and more, you tend to get this notion that you're going to even out. So let's look at a drift experiment. This is a really nice experiment. In this experiment, we have a population of flies, and we're going to look at an allele in these flies, which is either BW75 or BW. Doesn't matter what they are, but we're going to take populations of flies, small populations, that have an approximate allele frequency of 50-50 for these two alleles. We're going to put them in individual bottles, and we're going to start the experiment by letting these initial 0.5 frequency flies breed together. And then we're going to collect 8 flies from 8 males, 8 females from each of the bottles. And then we're going to take those, allow them to breed again, and just kind of keep doing the experiment. And in each generation, we're going to measure the allele frequency of BW75 and BW. So here's the experiment. The idea here is you have the number of bottles with a particular allele frequency. Okay, so some of these are higher, some of them are lower. So that's the number of bottles that have a particular allele frequency, BW75 and BW. And down here we have tabulated frequency of BW75. Right, so here it's half and half. When we start the population, or soon after we start the population, the flies in these bottles are spread out with an approximate allele frequency of half BW75 and half BW. All right? But as we collect 8 males and 8 females from each of the bottles, let them breed again, we see just statistically a kind of movement in allele frequencies off to the side. Right? So we start to see almost right away spreading of the allele frequencies, just due to the fact that we're just accidentally, we're kind of flipping a coin. Sometimes we're getting 8 heads, 2 tails, sometimes we're getting 8 tails, 2 heads. That's kind of the directionality that we're going here. Okay? So right away we move off to the side. If you already move over to this side or to this side, it becomes a lot more likely that you're going to continue to pick those dominant alleles over here or over here. Not dominant, but the more, the allele in higher proportion that's over here and that's over here, right? So you can start to move off to each side, and by the time you kind of get to these middle generations, and particularly at the end, you've kind of swung either one direction or the other direction, and you've completely lost one of the alleles. So these bottles of flies over here have an allele frequency of BW at 1 and BW75 at 0. The BW allele has been fixed in the population, and we've completely lost BW75. The flies in the bottles over here have fixed the BW75, and we've completely lost BW. And this is just due to small population, randomly picking organisms, and based on who mates with who in these bottles, and kind of the chance of picking flies of certain genotypes. So it's an idea of a drift experiment, and gives us this notion that in small populations we can change allele frequencies just through this kind of random process. Let's see, flies in this model mate, collect 8 male, 8 female. Okay, so there, I've said pretty much what's here. This is a neat experiment because you can just so clearly see changes in allele frequencies based on small population size, and you see the fixation of one allele or the other. More ways to change allele frequency. Bottleneck. Bottleneck is basically something bad happens. You have, let's say, a big starting population with lots and lots of different types of alleles, and then you squeeze the population, as shown by this narrowing here. And so only certain individuals are going to be able to pass through the bottleneck. Everything that originates and proliferates after the bottleneck has to have been derived from the alleles that managed to make their way through the bottleneck. Sometimes this might be directed, right? In the case of a disease, maybe it affects some organisms more and some organisms less, in which case you might have some sort of a skew in allele concentrations here that reflect that environmental pressure. Other times, if it's, a tidal wave or, volcano or something like that, you might think it affects everybody equally without regard to genotype, and what you get squeezed through the bottleneck is kind of a random sampling of whoever made it through, so to speak. These types of issues are important in conservation biology, where you have fewer and fewer organisms of a certain type, and in that case, you have far fewer alleles to select from to create organism diversity and so forth. So people worry about these types of things in really small populations. Founder effect is a somewhat similar idea. The idea of founder effect is that you start a population from relatively few individuals. So everybody who originates from those individuals are going to have the same kind of allele composition of the founders, whether that's good or bad. And the book has a really neat example. They describe all these kind of trials and tribulations of individuals from this island, Tristan da Cunha. And it's really interesting to read all the things that have happened to the people on this island. I always find it fascinating. And I have a picture. It's a great place, right? It's beautiful. You've got everything. So it's a small population. That could be a problem, a small population of founders. You've got beaches, right? It looks like a vacation paradise. But they've got three big problems. One is the population is really small. So everybody who is kind of originates here has to have been genetically related to those people who first founded it, maybe shipwrecked sailors or so forth. Okay, so that's one problem. The next slide shows another problem they have. That's a big problem, right? Do you see the problem? There it is. They've got a big problem right here. Here they are, right? And they've got a population size problem, a founder effect problem, and they've got this problem. And the book describes some issues about this. They have a third problem, this, right? This is the third problem, okay? It's a long ways away from anything. And I've got one more picture to kind of show where it is, and you lose sight of it. It's somewhere here, okay? They say it's about halfway between, what is this, Cape of Good Hope and Cape Horn. Is that Cape Horn? Anybody a sailor? So somewhere here. It's almost as far away from anything else on the planet that you could possibly be at, right? It's absolutely stunning. And I looked it up once. You have to take a ship from Cape Town, and it takes like 2 weeks to get there. So it is one remote place. So read the stuff in the book about it, because it's really fascinating. Okay, so how much time? We've got hopefully time for a couple more topics. So here we'll look at migration. Migration is one of the major assumptions that we have to worry about in the Hardy-Weinberg equation. We assume there's no migration, but now we're going to kind of treat migration. How do we do that? So let's consider an allele, big A, and we've got two populations, population X and population Y. And we're going to say that some individuals from population X with an allele frequency P of big A of 0.8 move into population Y with an allele frequency P of big A of 0.5. So now we've got two populations merging with individuals that came from populations where the allele frequencies were not the same to begin with. And we have to be careful about how we deal with these types of situations. Okay? Let's suppose population Y is 90 animals and 10 animals from population X move in. Okay? So really all we're doing is we're setting up a proportion. There's a new population size, and we want to know, what fraction of that new after migration population consists of animals from population X, and what fraction consists of animals from population Y. Okay? So the new population Y makes up 0.9 fraction, right? So 90 out of 100. And then in the new population, X makes up 0.1 fraction of this new population of 100. So we just kind of set up an equation where we take 0.1 times 0.8, which is the allele frequency of X. So they're contributing 10% of the new population, and they have an allele frequency of 0.8. and we take 0.9, because that's 90% of this new population, but they have an allele frequency of 0.5. We multiply them and add them together, and we get a changed allele frequency in this new population of 0.53. And 0.53 makes sense, right? It's higher than 0.5 because the animals moving in have a higher allele frequency for A, but it's not too much higher because only 10 animals moved in out of 90. So one thing you want to do when you look at these types of problems is just give yourself a kind of quick check to ask, does it make sense? Does it make intuitive sense? If you get something really weird, like it goes down, right? And then you think about, well, wait a minute, that can't happen. So make sure you kind of check in common sense whether things are working in the direction that you might expect. Okay. In picture form, it looks like this. Population 1 here, population 2 here. We combine them together, right? So the yellow makes up a certain fraction of the new final population, and the blue makes up a certain fraction of the new final population. And so we take the fraction made up by the yellow times its allele frequency plus the fraction made up by the blue times its allele frequency, and we get the new allele frequency in the combined population. Okay, so some notes about this. It doesn't have to be 2 populations, it could be 3. The process works the same, right? It's still the relative contribution of each of the 3 populations to the final times their respective allele concentrations. So that could be a variant of this type of a problem. And the other thing is to be very careful about applying or not applying Hardy-Weinberg in these types of problems. This population here, to begin with, may or may not be in Hardy-Weinberg equilibrium. Maybe the problem would tell you that, maybe it wouldn't, okay? But if it is, and the allele, if both of them are in Hardy-Weinberg equilibrium, and the allele concentration is different in the two, then when you combine them together, you're no longer in Hardy-Weinberg equilibrium. That violates that, the assumptions of that. So you would have to be very careful about generating a new allele frequency here and then plugging it into Hardy-Weinberg and getting genotype frequency. So you don't want to do that. Okay. So I think the last topic we'll talk about today has to do with changes in allele frequency through time as a result of these fitness constraints. So if you have a population of alleles in, let's say, generation 1, and the types of individuals that you could get out of that would be based on picking alleles out of the population of alleles at that snapshot in time, combining them together and making certain individuals. So the allele population at time 0, let's say, is finite, right? You have a certain number of genes, a certain number of alleles, and the only thing you can make in terms of individuals is based on selecting from that finite population of alleles that you have in the population. If that set of alleles and their frequencies never change after 100 or 1,000 generations, then it would be exactly the same. And if you did the same thing again, you picked alleles from that pool 1,000 generations later, and nothing had changed in terms of allele frequencies, the range of individuals that you could make would be exactly the same as well, right? You can only make types of individuals based on what types of alleles you have in that population. I mean, maybe the variety could be, immense, but nonetheless, finite number of alleles in the population, you draw them out, you can only make a certain range of individuals. If that remains unchanged over time, then you can only make the same types of individuals 1,000 generations later. We obviously know that allele frequencies change, and part of the reason they change is that organisms do not have the same number of offspring, right? If everybody has the same number of offspring, then things are pretty static through time. But the fact is, some organisms have no offspring, some organisms have a lot of offspring. That means the alleles from organisms that have no offspring aren't being passed on at all, and the alleles from organisms that have lots of offspring are passed on at much higher frequency than those that have fewer offspring. That kind of makes intuitive sense. So we can define this term called fitness, which is a little bit cartoonish in our example here, but nonetheless, there's a term called fitness which we designate by this variable W, and we can we can say, we can define a fitness for different genotypes. So here we have 3 genotypes, so it's a 2 allele system. Where's my pointer? G1, G1, heterozygous G1, G2, homozygous G2, G2. So these are 3 types of organisms that might be in some hypothetical population. And we observe that G1, G1 organisms have 8 offspring, G1, G2 have 4 offspring, and G2, G2 have 2 offspring. So this is just given to us in this example so that we can understand what we're doing with fitness. So the G1, G1 is more fit. More alleles are being passed to the offspring from G1, G1 individuals compared to the alleles being passed to the offspring from the G2, G2 individuals, right? So you can imagine that there's going to be a change in allele concentration of G1 and G2 just over 1 generation in this kind of crude example. This is going to be far more abundant in the offspring relative to the G2 allele. So the fitness is defined as the number of offspring divided by those individuals that have the maximum number of offspring. So these individuals have 8 offspring, let's say, and we divide that by 8, giving this a fitness of 1. Fitnesses are always out of a value of 1, so 0 to 1, okay? These are the most fit in our example. 4 out of 8 is 0.5, and 2 out of 8 is 0.25. So it's very crude, but it gives us a way to kind of look at allele frequency changes as a result of these types of fitness or selection problems. One of the problems that I see happen a lot is people will add up all the offspring, 8 plus 4 plus 2, and they will divide 8 by, what is that, 14? 8, 12, 14, 8 by 14, 4 by 14, and 2 by 14. No. You get the maximum here, 8, you have to divide by 8. Everything is divided by those organisms that have the maximum number of offspring. So be careful with that particular problem. And obviously this is an oversimplification. We're just doing this to illustrate kind of the type of thing that happens. We could imagine all kinds of scenarios like this is way too many and they can't take care of them and they all perish, so forth, right? That's going to throw this whole thing off. So we could imagine all those things, but let's not worry about, these types of things. Let's just take it for what it is. We have a fitness calculated for these, and for these. Okay. So in these fitness problems, typically what you're asked to do is begin with a starting population that has a certain genotype frequency for individuals and allele frequency. You're asked to apply these fitness constraints, and you're supposed to determine genotype frequencies and allele frequencies after you apply the fitness constraint. So that's kind of stated in words here. Start with the population, certain allele frequency, genotype frequency. Maybe that will be given, maybe you have to do some calculations. Apply some fitness constraint, the W. Maybe that will be given, maybe you have to calculate it from the number of offspring. You calculate allele frequencies and genotype frequencies of the resulting population, and you kind of then get a sense of how things changed as a result of applying that fitness constraint. Okay, it's basically plug and chug. There's a table that I'll go over next where you can follow this, and you pretty much just follow the table and you'll be fine, okay? Looks like a mess, but it's not. Let's see, things to keep in mind. I'll try to come back to this if I can. In the book, there's a lot more mathematical formulas and tables and so on and so forth, and the problems tend to get a little bit complex. Again, follow the lecture notes as a guideline for the kind of difficulty or the types of problems. I see a lot of formulas in the book, and I see people try to apply those formulas in some fancy way for these fitness problems, and we're really not dealing with a lot of that. And sometimes mistakes get made as a consequence of that, so just be a little bit careful. We're just looking at this basic framework, which is pretty straightforward. So there's a table here that describes how you do these, okay? And in this table, we start with a population with allele frequency P of 0.6, Q of 0.4. That's a given. We don't know that, it's just a given, okay? And we're gonna make the assumption, or we're told, the population is in Hardy-Weinberg equilibrium. And if it is, then we can use P squared, 2PQ, and Q squared to determine the genotype frequencies for, in this case, A1, 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 A2, and A2, A2. Okay, so we have to either, boldly do that, use Hardy-Weinberg, or we're told that we can do that, one of the other. So genotype frequencies, P 0.6, Q 0.4, we have these terms, P squared, 2PQ, Q squared, we just plug in the numbers. So P squared, 0.6 squared is 0.36, 0.48, 0.16. So far, we haven't done anything different than we did with the, let's say that cystic fibrosis example. We just have allele frequencies and we plug in to Hardy-Weinberg. So this means 36% of the individuals in our starting population are A1A1, 40% are A1A2, and, 36%, 48% are A1A2, 16% are A2A2. So great, so far we haven't done anything really. Now we apply our fitness constraint. Here, in this problem, the fitness is given to us. We haven't calculated it from offspring numbers. We're saying the fitness W for allele A1A1 is 0. Well, that's pretty dramatic, but anyway, that's what we're given. The fitness of the heterozygotes, this is interesting, it's 0.4, that's less than 1, and the fitness of the A2A2 is given at 1. Somebody's got to be 1, right? There can be more at 1, but somebody's got to be 1. Somebody has the most number of offspring, as defined by our kind of cartoonish fitness definition. Okay, so this is bad. What we do then is multiply the initial genotype frequency times the fitness value that we're given. So 0.36 times 0 is 0, 0.48 times 4 is 0.19, and 0.16 times 1 is 0.16. So now we get new genotype frequencies in a population that we've applied our fitness constraint to. Okay? And let me just kind of mention again, sometimes I get this question of where do these W values come from? In this problem, they just make them up. They give it to you. Okay, that's all it is. It could be anything, as long as it's between 0 and 1. Okay, so we apply the fitness constraint to the genotype frequencies. So here we have 0 frequency these, 0.19 for these, 0.16 for these. But this new population should be reset at a level of 100%, or 1, right? 0.19 and 0.16 don't add up to 1. So we have to normalize the initial genotype frequencies that we get by multiplying by the fitness constraint to a new population that's now a new whole, right? So we add up 0.19 and 0.16, and we get some value 0.35, and we have to compare these new genotype frequencies to our new population whole, which is 0.35. So nothing over 0.35 is nothing. 0.19 over 0.35 is some value 0.54, and 0.16 divided by 0.35 is 0.46. Now we have the relative genotype frequencies in the new population kind of reset at an out of 1 value. So that's important. Now, we may have changed absolute numbers during this process. The population may be smaller, okay? But the key here is that we've renormalized these things to an out of 1 situation. So it's not that hard. You do some initial calculations, you multiply it times the fitness value, you renormalize based on what's left in the population, and there you go. Okay? So this is one conclusion of these types of problems. What's the new genotype frequencies after selection? And the other thing that you normally expect to do in these problems is calculate new allele frequencies. And in this particular chart, what they've done is they've used that genotype frequency plus half the heterozygous genotype frequency, right? So genotype frequency of the homozygotes plus half the genotype frequency of the heterozygotes. That's one of those 2 approaches that we looked at for calculating allele frequencies. So here we have P prime, meaning the new allele frequency equals this genotype frequency plus half this, so that's 0 plus half of this, okay, which is 0.27, And we know the P and Q values, since it's a 2 allele system, have to add up to 1. So we just take 1 minus 0.27 and we get 0.73 for the new frequency of the Q allele. So done, we did it, okay? We started with the population, applied a fitness constraint, determined the new relative genotype frequencies, and then from those calculated new allele frequencies. So let's look at the new allele frequencies. What happened? P is 0.27 now, whereas originally it was 0.6. So it dropped dramatically as a consequence of selecting against it in this column right here. Q is 0.73. It shot way up. It was originally 0.4 because it is now, relatively speaking, a greater proportion or abundance compared to P than we had when we started, because we got rid of so many of the P. Okay, so this is about what you should do. I don't know if there's so many variants of this type of thing. Typically, you may be asked to, when should you apply Hardy-Weinberg and when shouldn't you? Should you at all? So you shouldn't worry about that, because this type of process usually violates this Hardy-Weinberg assumptions. Sometimes you might be given the W value, sometimes you might have to calculate it like we did over here. So don't mess this up, because if you mess this up, then you're going to mess this up. And I guess the last thing is, don't overcomplicate it with all those fancy formulas that they have in the book. Okay, last thing real quick. We can apply kind of hypothetical fitness constraints on various allele combinations relative to one another. In this case, we say fitness of alleles 1,1 equals fitness of 1,2 equals fitness of 2,2 equals 1. Everybody's the same. All the alleles are equally fit. There's no selection for or against them. That's one possibility, right? Let's see. I'll just do selection against recessive. Here we have the fitness of the recessive allele in homozygous form, 2,2, is less than 1, but the fitness of homozygous 1,1 or heterozygous 1,2 is equal to 1. This is typically the case of your standard recessive genetic disease, where there may be selection against being homozygous, but there's no selection against wild type dominant and no selection against the heterozygous genotypes. And you can kind of look at these other ones and kind of imagine how they would work. You can have all sorts of possibilities. The other, real briefly, selection for hetero. Here is something that might be the case for the sickle cell trait, or maybe that cystic fibrosis trait, under certain situations where you have some advantage if you are heterozygous. And this is one reason why kind of that allele might rise in the population. So this is selection for heterozygosities. These are kind of interesting to me. Okay, that's it for today. I'll see you next Tuesday. We'll finish up a little bit.